So the next of the 5 I's is incubation. So once you've done your inoculation and you've put the sample into the media so that it can grow, you'll want to put it in the incubator. And the incubator is basically this temperature controlled chamber that is going to encourage the growth of microorganisms. So typically with microorganisms, most tend to grow well at about 37 degrees Celsius, which is comparable to 98 degrees Fahrenheit, which is our internal body temperature. So if you let them grow at room temperature, they'll grow, but it's usually very slowly. To encourage growth at a fast rate within 24 to 48 hours, we will typically put them in the incubator at 37 degrees Celsius, and then with that temperature, we will have an abundance of growth within 24 to 48 hours. The next I is isolation, and isolation is a way that we can inoculate a plate and get what we call isolated colonies. So if we look at this plate, we have all these little isolated sections, these little circles. Each one of these is what we call an isolated colony. And a colony is a macroscopic cluster of cells appearing on a solid media, which we see here with the Petri dish, that arise from the multiplication of a single cell. So if we look at this single individual colony, this individual colony is where one cell was placed, and then that cell continued to grow and grow and grow until we could physically see a macroscopic cluster that we recognize as a colony. So there's not just one bacterial cell in this colony. This colony is actually one bacterial cell that over a 24 to 48 hour period became 2, and then 4, and then 8, 16, 32, etc., until it formed this visible cluster that we can see. The nice thing about having an isolated colony is it's ideal to pull a sample from, because if we pull a sample from this isolated colony, we know that every single cell in that colony has the same genetic makeup since it originated from a single cell that was deposited there 24 hours ago. And that is nice when we want to run other tests or put it under the microscope because it avoids that contamination factor. So ideally, when we inoculate plates, we sometimes need to achieve what we call these isolated colonies. And a technique that we could use to do that is something called a streak plate technique. Because unfortunately, a lot of times, if you just collect a sample of bacteria and then just put it in the Petri dish, you will get something like this over here. And it'll just be a big blob of bacteria with no isolated colonies, which often can hide contamination. But when we do specific techniques like a streak plate technique, we increase the odds that we're going to get isolated colonies that we can then pull samples from for further testing. So, how do we do a streak plate technique? A streak plate technique is basically a way that we can inoculate the plate and do dilutions at the same time to increase the odds of us getting isolated colonies. So oftentimes you'll be given a sample of bacteria. So this is a test tube. It has a nutrient broth in it, and there's bacteria growing inside. So it's a mixed sample because we have 2 different types of bacteria, the orange bacteria and the blue or purple bacteria. So to do a streak plate, you would collect a sample from the mixed culture, and with that sample, you would take the sample to a Petri dish, and on the surface of the solidified agar, you would streak your loop back and forth approximately about 10 times to create what we're calling quadrant 1. So that would just be in this top section here. Then you would take your loop and you would sterilize it. So you typically subject it to heat, like in an incinerator or the flame of a Bunsen burner. You would sterilize it to get any extra bacteria off of it. Then you would come in after sterilization and take a small sample of your quadrant 1 and pull it over to make quadrant 2. And then again, you would sterilize your loop. You would come back. You would take a small sample from quadrant 2 and pull it down to make quadrant 3. And then again, you would sterilize your loop, and then you would come back and you would pull a small sample from quadrant 3 over to make our final quadrant, which is quadrant 4. But collectively, what we're doing here is we're diluting the sample. We're putting less and less bacteria in each section so that these bacteria, once there's less of them, they'll have more space to grow so that they can mature in isolated colonies. So after we've done the streak plate technique, we'll incubate for 24 hours, and then we'll look at our final product. So in quadrant 1, we can see how the bacteria kind of all just grew together in one big blob, and we can expect that. Quadrant 1 is going to have the highest concentration of bacteria. There's not enough space for them to spread out, so they're just going to grow together in one big blob. Quadrant 2 has been diluted out some, but there's still a lot of bacteria there, so we still kind of get this merging of the bacteria. By the time, though, we get to quadrants 3 and certainly quadrant 4, we have limited bacteria. The sample's been diluted substantially. Those small numbers of bacteria have the space to mature into isolated colonies. So it'd be very easy at this point to grab a sample of purple and put it under the microscope, or grab a sample of orange and put it on the microscope. If your entire plate looked like quadrant 1 and I asked you to get a sample of orange, it'd be really hard to do that without contaminating it with the other bacteria. But by achieving isolation, it's very easy to go in and get the specific sample that you want to obtain a nice pure culture. And this is just an example of what a good streak plate versus a bad streak plate looks like. So ideally, we'd always want to get something like this. We know that quadrants 1 and 2 are going to be virtually unusable because while the sample has been diluted out in quadrant 2, it's not enough. So the high concentration of bacteria in quadrant 1 and quadrant 2 typically doesn't promote isolated colonies. But by the time we dilute it in quadrants 3 and certainly in quadrant 4, we get those nice isolated colonies that we can use for further testing. A bad streak plate would look something like this. In this streak plate, some common things that happen is maybe you collect a sample and you make quadrant 1. And then you collect another sample and you make quadrant 2. And another sample for 3 and another sample for 4. What you've just done is add 3 times as much bacteria than you were supposed to have originally. You only go into the original sample to create quadrant 1. All the other quadrants past quadrant 1, so 2, 3, and 4, are made as a product of pulling a small sample down from the previous quadrant. But you only go into that original quadrant, or that original sample, to create quadrant 1. It looks like what might have happened in this plate is this person forgot to sterilize their loop in between quadrants. So they probably did quadrant 1, and then immediately pulled from 1 to make 2, and then 2 to 3, and then 3 to 4. Well, if you're not sterilizing your loop in between each quadrant, you're not getting the extra bacteria off, which means you're basically just stirring your culture around. So you do have to sterilize in between to get that excess of bacteria off. But the whole goal with doing a streak plate technique is to achieve isolated colonies so that you can use those colonies for further testing. The next two I's relative to microbiology labs are inspection and identification. And while you need to know that these are part of the 5 I's, I don't need you to know the detail as much as you did for inoculation, incubation, and isolation. Inspection and identification have more to do with things that are relevant in the actual laboratory, so we'll be covering these more in lab. Things like, what do bacteria look like when they're under the microscope? What's their shape? What's their arrangement? What do the colonies appear like when they're growing on a plate? Do they interact with the media in any way that indicates they utilize special nutrients or go through specific fermentation processes? So these are part of the 5 I's, inspection and identification, but you don't need to know the details pertaining to them as you'll get that experience in lab. Now moving on to microbial size, I don't need you to know any specific measurements. I just want you to understand that microorganisms are microscopic. you cannot view them with the naked eye. They do require the use of a magnifying device, such as a microscope. As far as relative size, microorganisms are about, the larger microorganisms are about 10 times smaller than what we would see for like typical human cells, at least for bacteria. Larger microorganisms, things like fungi, are comparable to human cells, but bacteria, as we typically discuss, they're about 10 times smaller than eukaryotic cells, such as human cells. So really with things like fungi and protozoa, they're the larger of the microorganisms. Bacteria are kind of in the middle, and then the smallest of the microorganisms are viruses. Viruses are exceptionally small. We have to think if the influenza virus infects one of our respiratory cells, it can replicate and form about 1 million influenza viruses. So if 1 million influenza viruses can fit into one of your respiratory cells, you can imagine how small viruses are. So this link here is really great at showing you relative size. Again, there's no specific measurements you have to memorize, but it kind of just shows you, in comparison to human cells like red blood cells and white blood cells, just how small bacteria are, and then how small viruses are compared to that. But just have a general understanding that microorganisms are exceptionally small and require the use of a magnifying device in order to be visible. So speaking of the magnifying device, our next topic is light microscopy and the use of a compound light microscope in order to make specimens visible. With light microscopy, there are 3 essential principles that we'll be going over. The first being magnification, which is the ability to make an image larger, which is what we need to do for microorganisms because they are so small. The second is resolution, which is the ability to see the image clearly. It does us no good if we magnify something and it's blurry. We need to magnify it and also achieve clarity or resolution in order to get a clear image. And then contrast is the ability for the specimen to stick out against its background. So if we look at this slide here, it has a white background right here with black text. So that's really good contrast. It's easy to view the text. However, if I would have picked a light pink font against a white background, you could see where the contrast would not be good, and it would be very hard to visualize the text. So as far as lecture is concerned, you don't have to memorize the parts of the microscope or their functions. That's something that'll be utilized or done in lab. But as far as lecture, I do want you to know the pathway that light takes as it travels through a light microscope. So it's fairly easy. It's just an order of 6 steps. The light starts out at the light source, which is essentially just the light bulb. From there, the light is projected upward from the illuminator, kind of like a flashlight projecting the light upward. When the illuminator projects light upward, a lot of it is kind of fanned out with scatter. So the next stop is the condenser, which condenses or kind of collects and focuses the light so that the light is directly projected to the specimen, which is whatever you're looking at on the microscope slide. From the specimen, the light will then go to the objective lenses, whatever one you have clicked into place. Now these objective lenses produce the first initial image of the specimen, called the real image. From there, the light will then travel to the ocular lens, which is in the eyepiece. This is going to generate a virtual image, which is the magnified image that is perceived by the brain. So you will need to know these 6 steps as far as light passing through the microscope, and then, of course, which lens system produces which image, with the objective lens producing the real image and the ocular lens producing the virtual image.